Vince Calhoun, thank you very much for coming online today to give this talk. Uh, Dr. Calhoun is founding director of the Tri-Institutional Center for Translational Research in NeuroImaging and Data Science. And just before this, we were having a conversation, and he told me that his aim was breaking the silos around Georgia Tech, Georgia State, and Emory, and that's really what we believe in. So that's what really is the future of the science. He is a Georgia Research Alliance eminent scholar in brain health and image analysis. He was previously the president of Mind and Research Network and distinguished professor of electrical and computer engineering at the University of New Mexico. He has authored more than 900 journal articles and 850 technical reports, abstracts, and conference proceedings. His work includes development of flexible methods to analyze functional MRI data using tools like ICA, deep learning for neuroimaging, data fusion for multimodal imaging and genetics data, neuroinformatic tools, and the identification of biomarkers for the disease. This is where I think science is going, where we are able to collect multimodal data from all possible things. The major challenge that remains, some of the issues that we will address today, is how do you integrate all this together so that it forms a coherent picture. He is a fellow of IEEE and AAAS, the American Institute of Biomedical and Medical Engineers, the American College of Neuropsychopharmacology, the Organization of Human Brain Mapping, and the International Society of Magnetic Resonance in Medicine. He has been on a large number of professional committees guiding many professional organizations. I'm most pleased, and as all of us in the School of Eight, to welcome Dr. Vince Calhoun for this Thar talk. Dr. Vince, it's all yours.
All right, great. Well, thank you. Well, it's great to be here virtually. What I want to do today is talk about data fusion of neuroimaging data. It's something that's talked about a lot, but it can be challenging to do, but there are some actual very real benefits to be had. So I'll just give kind of a brief sort of overview of some introductory topics, introduce some methods, and then I'll talk about a few applications of those methods, and then some additional models. And depending on time, we may cut that a little short. We'll see. So essentially, multimodal fusion, what you're trying to do is capture complementary information between modalities. We have structural, morphometrics. We have diffusion, with sort of structural connectivity, electrophysiology, brain function, functional connectivity. You could bring in things like DNA expression or DNA methylation or single nucleotide polymorphism array data. There's lots of information that you could bring in there. And you can think of fusion as sort of a spectrum ranging from just do separate analyses and look at them side by side to sort of integrating, like maybe trying to co-register MRI to PET data and look at them overlaying on one another. Asymmetric fusion would be maybe I use fMRI to provide seed regions that I use then to model or inform an EEG source reconstruction model. And then symmetric fusion would be allowing the modalities to sort of jointly inform the model. So that's sort of the highest information case. You can also think about late and early fusion. Late fusion would be I do a bunch of processing and then I combine the information after that fact. Early fusion would be to sort of keep things, a little bit more raw and combine early on. And those have different sort of advantages depending on what your goals are. As I mentioned, we're trying to look at this joint information. So if, say, for example, we were trying to differentiate groups, you can conceptually see, well, maybe brain structure and brain function, neither one shows a detectable group difference in the distribution. But if we were to draw a line through the modalities like this, we could start to see the distribution separate. So that's the joint information that we don't have access to if we look at only one of the modalities. Similarly, I do a lot of work with sort of multivariate analyses, looking at patterns in the data. And so instead of looking at just sort of univariate relationships where we sort of look at point-wise, correlations or dependencies in the different data sets, we want to sort of capture whether there are patterns that potentially co-vary or have other relationships between the data sets. So the use of data fusion approaches has been increasing, and as you can see, sort of in this publication chart, and it is pretty clear that it improves performance in a lot of scenarios. So, for example, here's prediction accuracy looking at multimodal versus unimodal studies, and you can see that the distribution here is shifted to the right. So there's a higher accuracy generally when you're looking at multimodal information, and presumably because you're capturing complementary information, leveraged across the different modalities. I often try to think of this in the context of sort of, should we do fusion? Think about it as a carrot and a stick. So the carrot is, well, if you combine, say, MRI and EEG, we can see high resolution spatial information from fMRI, and we can see temporal information from EEG at high frequencies. And so we kind of get the best of both worlds. The stick would be, well, if I look only, say, at fMRI data here, and compare that with MEG data, so you can see that the MEG data is showing a lot of, first of all, sort of differences from the fMRI data, but also between, say, these two different stimuli, novels and targets relative to the fMRI. So what is the right answer here? I would argue neither one of these is the right answer, but they both are providing a window into the underlying phenomena we're trying to study. And so we should be looking at both of these. MEG is emphasizing very different things. It's looking at sort of magnetic fields generated by bundles of neurons at high frequencies. fMRI is looking at blood flow, blood oxygenation. Okay, so one of the things that we leverage a lot in data fusion approaches is inter-subject covariation, and generally by incorporating high-dimensional features. So a feature is basically something that we construct from a data set. So for example, with fMRI data, we have a 4-dimensional data set. Maybe it's a task. In the context of a task, we could compute a task activation map. So that's just one example of it's a high-dimensional feature still, but it summarizes the task relevant information. Similarly, you could compute a functional connectivity map. For structural MRI, maybe instead of using the raw data, I want to just focus on the gray matter portions. And so I might want to do a segmentation there. So this gives us a summary of the data. It's less complex, potentially easier to model, and yet we are preserving a lot of the dimensionality of the data and being able to move forward. There's still a lot we can learn there. So we're not reducing down to, say, a single region of interest. And just to kind of show you how much we can, how much information is contained in this covariation. If I look, for example, at task maps generated from, an fMRI task. In this case, I think it was an auditory oddball task. And I simply look at covariation among subjects in those task maps by using independent component analysis. I apply independent component analysis to this subject by voxel matrix. And what do I find? Well, if you look here, these are the different components that we identify. And what I'm showing you on the left here are corresponding resting fMRI networks generated from the 4-dimensional fMRI data. And then these are the corresponding covarying patterns that were identified from the task maps. And you can see how similar they are. What this is telling us is that the resting network information that we typically look at is modulating the task-based information across subjects. And so we can still extract those, those networks by looking at this covariation. And so we can use this as a way to sort of link data sets, because oftentimes subjects have multiple data sets, multiple modalities collected. So I might have the same subject as my linking variable and multiple types of data. Another example where we've used this is to look at the correspondence between brain structure and brain function. And so we can take gray matter maps and again, look at independent component analysis of that data to get out patterns that show patterns of gray matter that covary among subjects. And so if we do that, we get patterns that look like this on the left. Corresponding on the right here, I'm showing you resting fMRI networks extracted from data collected from the same subjects. And we've sort of matched these. And you can see that the gray matter information is actually also reflecting patterns that resemble what we see in these resting networks. Whether that's due to actual gray matter volume variation among subjects or to some information in the MRI signal that's preserving function in the gray matter maps, I think is kind of an open question, but it's still generally what we find is the gray matter information is sort of less specific, and we find often multiple networks in resting fMRI that match to one in gray matter, for example. And we can use this kind of information sort of side by side to compare, even very different modalities. In this case, fMRI and MEG, magnetoencephalography, where we've basically done essentially a group ICA on the two data sets and then matched the corresponding spatial patterns. And this is spatial ICA done in both cases. So the MEG data tends to be a little bit less spatially focal. Resolution's a little bit lower, but we still find, again, you can see here, for example, visual components that fall within visual networks. They don't match exactly to what we find with the fMRI data, where we get sort of a lot more granular information, but it gives us a way to sort of link and compare these different modalities. Similarly, we can look at diffusion data. So this is looking at fractional anisotropy. And so this is sort of a voxel-wise map that tells you the degree to which, the diffusion is occurring, in one direction, or I say isotropic all directions. And we can again look at covariation using ICA and find patterns that covary that can map onto existing known tracks in the data. And so I've got those color coded here. These are the different components that we identified. You can see they sort of fall along some different tracks that can be identified with atlases. However, this is pretty distilled from, this loses the directional information in the diffusion data, and we can preserve that by doing probabilistic tractography, where we essentially take a matrix of voxels, and we have a sort of all-to-all voxel matrix, and we can compute the probability or the number of times, that if we were to start in this voxel, we would pass through one of the other voxels. And so that gives us a map, a probabilistic tractography map, voxel by voxel. We can also evaluate covariation in that in the context of a group ICA model. And if we do that, we find components that map very nicely along directional information in the diffusion signal. I've kind of showed them in a rendered view here. It's a little easier to see. And so the components are separating along different directions. And you can say this is just a subset of the components within the corpus callosum. And so blending those two together, we can look at basically a combination of this diffusion information and functional connectivity, where we compute an all-to-all connectivity matrix, and we sort of link these two in a joint model. And if we do this, we actually find, you can see here, linked pairs where you get the diffusion information I'm showing you here in blue and the functional information in red. And here's another example. I'm showing you just one of many pairs like this that we find. And so it gives us this really, I think, nice way to link together diffusion and function in a way that is completely atlas independent. It's just it's completely data driven. So you can then impose, atlases after you've identified these patterns in the data. So in terms of methods, there are a lot of ways to do data fusion. It's really, it can be overwhelming if you're starting in the field. We've caused some of the problem here because we've developed a number of different methods with different advantages and for use in different scenarios. But a lot of these are sort of matrix decomposition approaches, a lot of sort of canonical correlation analysis or independent component analysis, or combinations of those. I'm going to focus on sort of one model within that family. I'll start with just joint ICA, which is a very straightforward analytic model where we're essentially assuming a common mixing profile, a common loading parameter. So here we have subjects in this direction, and we have the data points from modality 1 and the data points from modality 2 simply concatenated. And so what we're doing is identifying maximal spatially independent components that share a common loading parameter. So it's a straightforward model, but it can be really quite useful. It's one of the things that the results from this are really convince me of the value of data fusion. Here's the example I like to use. It's an older one, but we have two different tasks that we were run in an fMRI scanner, a working memory task called Sternberg working memory, and an auditory oddball task. We generated task maps using the task timing information, through kind of a standard fMRI analysis pipeline. And then we applied joint ICA to those task map pairs from individual subjects. We extracted the components, and what I'm showing you here is a component is sort of the joint distribution from a component that showed group differences in this case, schizophrenia patients and healthy controls. And what I found pretty interesting here is, if you look at these are on the left side, these are individual subject distributions where I've got auditory oddball versus Sternberg. Notice how the patient information is kind of tilted, whereas the control information is very flat. This is telling us that the joint information, or there's sort of a correlation between the patterns that are identified in the patients. Another way of saying that is that the activation profile in those patterns, reflected in those patterns, is more similar between the two tasks in the patients than the controls. Patients are sort of activating their brain in a more similar way to these very different tasks. Controls have more unique patterns of activation. So this is really significantly different. It's a very striking difference, but it's completely invisible to you if you only look at one of the tasks and you don't look at them jointly. So that was one of the reasons that sort of convinced me that data fusion, was a real important direction to pursue. Here is another example where we looked at fMRI and EEG data collected from the same subjects, but in different sessions. So it wasn't concurrently collected. We applied a joint ICA to event-related potentials computed from the EEG data for an auditory oddball task, and the fMRI activation maps, again, computed for the target stimuli. And so we're looking at covariation among voxels in the fMRI and time points in the event-related potential data. And so I'm showing you two examples of joint components where the yellow curve here is sort of the average ERP, and the blue curve is the component time course, and it's linked to this spatial pattern for fMRI. Here's another example. So this one kind of, sort of well captures the N1 peak in EEG. This one captures N2. Now, once we have these pairs, we can reconstruct a spatial temporal model from them. So for example, we can pick any point in time here, and then we have weights corresponding to this blue curve. And we can use those weights to reconstruct a combined fMRI map. And so that's what I'm showing you here in the bottom for different points in time. And this again was very striking to me because we see, if we wait till this to reset, you see early on, you see bilateral temporal lobe, then you see somatomotor, motor planning, motor execution, and then sort of a large, sort of P3 response. So this gives us a spatial temporal movie at the level of milliseconds from fMRI data, but we can only get that because we've linked it in this data fusion model to the EEG data. Okay. This is just one slide to comment on, I think that there's still a lot of work that needs to be done in simulating and validation in this kind of work. And it can be challenging to simulate even just one modality, but then you have two, and then you have the joint information. So that can be complex. One way that Roger Silva in my group has proposed to do this is using something called copulas, which is sort of a bivariate distribution. And there he's proposed an approach where he simulates this joint information, and then we can compare to the ground truth using the models that we run. Okay, and then just building on the joint ICA model that I mentioned, and I'll give some examples of using this model here in a second. With joint ICA, one of the challenges is that as you try to scale up modalities, for example, you can start performance can start to get diluted. This is related to the ability of the model to sort of identify the covariant patterns in this sort of higher dimensional space. One of the ways we can sort of boost that is by combining it with something called multi-set canonical correlation analysis. So these are sort of different models. So MCCA is really trying to take, say, sets of matrices and find maximally correlated, pairs, within those matrices. So it's kind of jointly looking at covariation. And it works well in that goal, but can suffer if there are multiple high, if there's not sort of distinction or diversity in the degree to which these patterns are covariating. Whereas joint ICA is very good at identifying maximally independent sources. And it assumes also that the loading parameters are sort of in common. So it gives us a common framework to evaluate. And so we can combine these two models together, essentially doing MCCA to sort of find the linked pairs, and then do joint ICA decomposition subsequently from that. And a sort of extension of that is something called guided fusion, or again, multi-set canonical correlation analysis with reference plus joint ICA, where we can take an external variable like, working memory or symptoms and guide the fusion analysis. And so it gives us a way to sort of focus the results. So let me just give you 3 examples. I'm just going to give you a couple slides for each. First one is looking at ECT treatment to individuals who are depressed. We're looking here at fMRI data, which was reduced down to a fractional amplitude of low frequency fluctuation map and gray matter maps. So we have brain structure and brain function. And what we did was we took a relatively large data set and we applied fusion with reference using the response information, in this case, the depression score, to guide the fusion. And then we evaluated the relationship between the responders and the non-responders in terms of their patterns that were identified. And so when we do that type of analysis, we find that treatment response has a very particular pattern of function and structural change. So for example, you can see here, this is sort of the average over all subjects. The left side is the functional change and the right side is the gray matter change. And you can see here in the responders, for example, the gray matter maps are showing changes like thalamus, caudate, hippocampus, etc. And the FALF maps in the non-responders are showing some changes here that are very distinct from these sort of frontal regions that are identified in the responders. And this was also predictive, of post-ECT responses in independent data. So again, it was, it gives us a way to sort of look at complementary information across these different modalities. Another example of that is to use, use cognition. In this case, we're taking schizophrenia data and we're applying it to 3 different data sets: fractional anisotropy again, fractional ALF, and gray matter maps. We're using our fusion is essentially a composite cognitive score collected from all of the individual subjects, and we apply this to multiple data sets. We essentially, in order to cross-validate the results, we basically identify a result, then we fix the maps and we go to the new data set and we reconstruct the relationships using those fixed maps that were trained on data set 1. And so what we found was sort of a remarkably similar pattern of gray matter and fractional anisotropy across different scores. So we have this sort of overall score, working memory, attention, and learning. But if you look here at the functional change, it's much more variable. You can see here we've got the composite score. You can see very different patterns in the working memory, the attention, and the learning. And that's also reflected in the bar graph here, where the fMRI components are reflected, 2 of them are reflected here in pink and yellow. And they vary quite a lot depending on what score that we're evaluating. And so this isn't necessarily surprising, in that functional information is probably going to be more reflective of what they're doing immediately in terms of their performance on a task or on an assessment. But it gives us a nice way to sort of link, again, these structural patterns, which are consistent, and they're consistent markers of this disorder, linked to these functional patterns. And the last example is looking at sort of subtypes and how do we evaluate heterogeneity. And so this is looking at an autism data set, the ABIDE 1 and 2 data set, and we're looking at the Autism Diagnostic Observation Schedule, the ADOS score, as our reference, and we're using FALF and gray matter maps guided by that ADOS score. Once we decomposed the data into these sort of components, joint components, we evaluated their relationship to existing subtypes that were present within these data sets: Asperger's, pervasive development disorder, and autistic subtype. And this, I'm showing you the relationship in sort of the initial model and in the cross-validated model. And so interestingly, when we looked at the relationship of function and structure, and here this is a little complex, but functional MRI is in blue, gray matter is in green, and linked FALF and gray matter is in red. And you can see each of these subtypes has a very different pattern of functional and structural covariation, and they're also linked to different subscores. For example, in this case, social interaction. In this case, social communication and social interaction. And in this case, those 2 plus stereotyped behavior. So again, it gives us sort of a nice way to evaluate the relationship, but also to look at subgroups. And you can do a lot. There's a lot of analyses you can do related, depending on what your question of interest is. So I'm going to just go maybe through a few of these. I'm not sure how much more time I want to spend, but I'll just give you kind of a couple additional models. There's so many different ways, to do it. And I wanted to show you a ton of stuff, but, there's a limit to what we can do in one talk. Here is an approach we call metamodal fusion. And so this is essentially, it's kind of similar in some ways to like what's called a radiomics approach, where you sort of throw in lots of different measures but these are higher dimensional measures. They're not, you know, just single scores or averages, for example. And then we look at the relationship within a Markov modeling space between each of the features and their subclusters. And so this gives us a way then to look directly at the probabilities between these different features, like, for example, feature 4 and feature 6. One of the things that's really nice about this framework is we can also look at multiple links. So we can look at maybe a case where feature 4 is associated with feature 2 through a relationship with feature 6. So here's an example where we have this particular feature is showing a relationship with gray matter, which then shows a relationship with brain function. So then we can look at sort of multi-paths. This is an example of two, where we have two-step path. And if we look at these relationships, and the details are in this paper, you can see for a lot of different types of information, structure, diffusion, fMRI, cognitive symptom scores, there's a lot of, you can see fMRI has captured a lot of the variation, but we also see structure, we see cognitive differences, and these are relationships to schizophrenia diagnosis, for example. One of the challenges in linking fMRI and gray matter, is most of the time you have to reduce fMRI down to a single map, right? So you lose the temporal information, you make some strong assumptions. So there are various ways to capture information in the case when you have what I'll call mismatched dimensionality. So we don't have a time dimension in the gray matter data, but we can link it using models. This is a deep learning model where we sort of have a sequence predictor and an attentional network. So it's a recurrent neural network model. And we're essentially linking together gray matter concentration maps. So the analogy is these are kind of like a database of images, and dynamic connectivity states, which would be something like the caption on the images. So there's a time sequence to those events. I'm kind of glossing over the pre-processing steps here, but we're using something called time varying or time resolved functional connectivity. And so when we use this model, we can identify links between structure and function. And we find very strong links. So these links are all well above chance, way above zero here. But we also see differences in some cases between the groups. So this is schizophrenia and healthy controls, very significant difference in this insula region. This is a gray matter pattern linked to changes in state 2, which is shown right here, which is a functional pattern of transient functional pattern of change. Okay. Let me skip this one in the interest of time, and I'll do this last one here, which I kind of think illustrates an interesting point. So again, trying to get at how do we capture the full temporal information in fMRI and try to link it to brain structure. Well, one thing we can do is, and this is kind of late fusion, where we might run a group ICA model on the EEG data and a group ICA model on the fMRI data, and capture the resulting information and then link it across subjects, as best we can. And that's sort of a parallel group ICA type approach. But it's a little bit, there are potentially advantages to allowing the data from one modality to be involved as you're decomposing the other data set. And so one of the ways that we've proposed to try to do this is something called joint parallel group ICA plus ICA. And so that's a lot of words, I know, but essentially what we're doing is a group ICA analysis of the fMRI data and a group ICA analysis of the structural data, but we're not just running it one time. We're actually running that decomposition And then we're identifying a linking relationship by looking at the similarity of the spatial patterns in the fMRI data, in this case to the group mean, and the loading parameters in the gray matter data. And so we're looking at that similarity and adding that as an additional constraint on the algorithm. So we essentially are iterating between these three parts: group ICA of function, group ICA of structure, and this linking term. And in the end, we get a model that gives us, component maps from each of the modalities. Here I'm showing you an example of the two. But we also have the time courses, the temporal information from fMRI in the model that we can use to construct functional connectivity matrices, for example. And so once we have this sort of linked model, we can then start to look at its relationship to various scores of interest. In this case, we're showing working memory, verbal learning, composite cognition in the two different groups. Okay, so in summary, where are we going? I think, obviously there's a lot of ways, to do this, but there's still a lot we haven't done. In particular, I think there are some data sets which can be very complex and probably require more nonlinear models. And so that's where deep learning approaches can play an important role. One example is linking of brain structure and genomic data or genome-wide data. So here we show an example where, looking at structure only, looking at a single nucleotide polymorphism array data only. So structure kind of separates groups a little bit, SNPs not so much. But if we do a joint analysis of the SNPs and the structural data, we start to separate the data, better than we had done if we just use structure. And then if we compute relevance maps from the network, we can see, well, which regions were involved in the gray matter portion and which SNPs here were involved in which pathways were they in that decomposition. I think for when you're looking at simply just trying to do prediction, for example, say from a 3D brain network, then that's where I think deep learning models can have play an increasingly important role. Here's an example where we're looking at Alzheimer's disease and progressive versus stable mild cognitive impairment. And so this is gray matter, but this is gray matter combined with function down here. I'm just going to focus on this one bar graph down here. And what you can see is our sort of, this is our accuracy of predicting the stable versus progressive MCI. And you can see that the performance is actually higher on the structural MRI than the function. But when we combine structure and function, our performance goes up even higher. And then just this last slide is just to say that, obviously, what are the, there's this dizzying amount of decomposition, matrix decomposition models now. And I showed you that slide earlier. We've been developing, again, this is Roger Silva, a unified model, something called multi-data set independent subspace analysis, which can implement all of these models sort of shown here. So joint ICA, group ICA, MCCA, et cetera. And so we kind of have a unified model with a unified cost function, and then now we can do more direct comparisons between these different models, because it's not particularly helpful to have many models that change in very small ways that give you very different results. So we really want to try to harmonize as much as we can across models. And then we can extend this framework to move beyond linear relationships. For example, we can add deep subspaces, for example, within this model. Okay, so I'll just summarize that. What I hope I've shown convinced you is that multimodal analysis, quite often provides benefits, a lot of benefits over unimodal data analysis, and gives you a window into otherwise invisible information. As I've mentioned, there's a lot of ways to do this. So I think, while we continue to develop models, we also need to think about how to harmonize across models. Flexible and unifying approaches can help with that. As I'd mentioned, the MISA framework. And then the fourth point is sort of by handling mismatched dimensionality, we can go further back and minimize information loss. So if you're getting into this, I would say start simple and then work your way up to more complex analyses, because it can be very challenging to start with a really complex problem if you haven't done it before. And finally, we've only scratched the surface, and there is a lot more that we've got yet to do. We try to release our software open to the community. So here's some examples of the tools. We do a lot of ICA analyses, as I mentioned, and those are all of that is contained within these toolboxes. And I want to say thank you to the center and to the many folks who contributed to these projects and did a lot of the work. So with that, I will stop. Should go ahead and stop sharing my screen, I guess.
Well, thank you, Dr. Khan. That was very exciting, actually. And maybe I will start with some questions before I ask other panel members to ask the questions. Now, this is certainly a nice way, once you have combined multimodal data and then you're trying to, say, differentiate between progressive dementia, say, in Alzheimer patients. Now, coming down to the reality of particularly a country like India or even in the United States, you can't really collect multimodal data from different patients. So the use I see it is that you collect multimodal data in an experimental setup and come up with what is the best for diagnosis or prediction or whatever you have for different mental disorders, for example. Now, is there anything that you have picked as your favorite, other than clinical observation? For example, you could do just functional imaging or just EEG, which is much cheaper, or something else. Is that something that you're looking at?
Yeah, so I guess I don't have the answer for you, but I can say that that is a very important question, and you're kind of getting at not only a practical issue but also an economic issue, and so I think there's so for example, we have 10 different modalities and each one takes an hour to collect, and some of them require certain equipment that may not be available locally. How do you decide whether it's worth collecting, say, this modality versus that modality versus both, right? And that's going to have to be done through a more systematic sort of, and we've done this a little bit, in the context of some schizophrenia studies where we're looking at brain function, brain structure, diffusion, different tasks, and asking, sort of, you can rank them in terms of not only unimodal, predictive power, but also joint predictive power. And so if your joint predictive power is about the same as, say, your fMRI or your EEG, or almost the same, then it may be worth, in terms of cost benefit, to just collect, the EEG. So those are really important issues. We're not directly focused on that sort of a comprehensive type analysis. We have looked at it a little bit, as I mentioned, but it's really important. And it's also going to depend on what you're studying, because I can say that, in the context of the work we've done, we've looked at, for example, Alzheimer's and schizophrenia, and we're seeing, fairly consistently, Alzheimer's is predicted, at least if we're looking at static measures, it's the predictive power of gray matter atrophy is quite high. Whereas if you're looking at schizophrenia, the predictive accuracy of functional connectivity is quite high, and vice versa. It's low in the other modalities. And so it's going to depend on the disorder, the question, and the modalities you have available. So that's a question that can be solved, that can be studied and quantified, right? So hopefully someone at your group can do that and give us the answer. So
yeah, thank you. So one more question. You passingly mentioned about integrating genomics data. Now that's another multidimensional, and for most of the multi-gene diseases, a highly diffuse sort of a data, if I would say. Have you attempted that?
Yeah,
integrating with the model? Yeah.
Yeah, I mean, we've looked, for example, we have, there was that one slide I showed where we were looking at structure and genome-wide SNP data. We've also looked at, and essentially, we've taken a little bit of a, there's different ways to approach it. You can start from sort of a blind approach, and you can say, let's decompose this data, and then after the fact, we can map it into, what are the putative pathways, bring in the bioinformatics databases, et cetera. Or there are other approaches where you can sort of try to inform the model that you're fitting, based on what you know about the genes and about their relationships, and about which pathways they contribute to. And so they have those approaches have different trade-offs and advantages. I can say that one of the we've looked at single nucleotide polymorphism data and DNA methylation probably the most. Those are the two we've looked at the most. The effects are very diffuse and small, and you need large studies, but they can give you some information about, especially if you integrate it with some of the large efforts like the psychiatric genomic consortia, where they get hundreds of thousands of data sets, and you can use that to sort of identify here's the set of risk SNPs, but then let's look at the multivariate relationship between those and the imaging data. And so that's kind of the approach that we've been taking. We found some interesting links between dynamic occupancy of a certain functional pattern in fMRI data and the polygenic risk score, for example, for schizophrenia. So that's one example of a result that we found that I thought was pretty interesting. So
yeah, thanks. Let me move on to other panelists, and then I'll come back with some few more questions if there's time. Dr. Anil Tiwari.
Professor Jain, thank you so much for giving me opportunity. Professor Calhoun, thank you so much for your very nice presentation. It was really very, very interesting to know the way you explained it, that when multimodal data is there and we are fusing it, then the information that we shall be getting out of this fusion will be more than the individual informations. But
Dr. Tiwari, if I may interrupt, can you turn on your video maybe?
Oh yeah, so yeah, sure.
Yeah.
Yeah.
Sorry, yeah.
Yeah, no, thank you very much. Am I visible? Yeah. I mean, I don't know.
Yeah, you are visible.
Yeah, thank you. So,
I like your hat.
Good evening from here, but I think it's a good morning at your end, most probably. Yeah. So it was very nice presentation. I was actually enjoying looking at each slide and then references you had provided at the bottom of it. Just curious to understand, in case you can provide us a few, maybe 2, 3 very important publications wherein we can pass on those things to students who wants to actually do research in that area. And because reading maybe many might not be easy, but then they are all very informative. This is one thing that I would like to request you, in case you can provide. And second thing I want to know, in case we can have some, or you can suggest us some depository where various codes are available, so that something quickly can be done, experimented, and people can find it more interesting. Sometimes it happens that when you have multiple models and when you are fusing it, then the probability function of individuals, right, when they are mixed, probably the mixed source, its probability is not predictable. So we need to have those things, separable kind of thing. So I have 2 questions. One, that 2, 3 very important publications you would like to suggest to students to read, and some codes.
Yeah. So there's one that we did a few years ago called, I think it starts with the missing link. And I could that kind of does a review of a lot of the methods that I talked about, and cites some of the other papers out there that are doing multimodal analysis. So that would be one. in terms of code, we have a lot of our code was referred to on that last slide. And those are toolboxes. We have MATLAB toolboxes and Python code, and they're all fully, they're pretty well vetted. So we even have sample data sets, and we have open code. And so you can run the toolbox and run the method very quickly. And so if you want to try it on some data, then that's available to do that. Let's see, did I get? And then the other point is, yes, and I was trying to be careful in saying that when you do multimodal analysis, it generally gives you a benefit, but it's not a requirement, right? Because obviously there can be some cancellation in some cases, or noise or other things, right? So but I would say that when we reviewed the field, generally the distribution was shifted to the right for multimodal analysis. So on average, it looks to be helping out, but it doesn't always. There's a spread. So
that's right. Because what I was thinking is that if there are orthogonality in the information from the various sources that we have, then when we are fusing it, we shall get additional informations. But no, at times, if we are not very careful, and then we are fusing some redundant informations, probably the benefits that we are expecting might not be that prominent, because we have not taken enough care.
Right.
Thank you, Professor. Thank you so much for responding and replying to my queries. That's all from my side, Professor Jain.
Okay. Thank you, Dr. Tiwari. Before I try to find out if Dipanjan is online, Dr. Rajendra, you have any question? No? Dr. Manish? Okay.
Yeah, so Professor Vince,
go ahead, please.
I just had one question. So thanks for your excellent talk. Towards the end, you had mentioned that deep learning could be sort of deployed here, right, for bringing nonlinearity. So I was wondering if bringing deep learning here, we might lose the visualization. Like initially we had PCA, IC, and all those techniques, right, which were linear and we can sort of visualize. So do you think that it might be an issue that the visualization part might go away as soon as we go to these deep learning models? So yeah, just your thoughts on that.
Right. So it doesn't go away. It's just harder. But it can be more powerful, I think. So I think, this is sort of getting at explainability and, and the visualization. But for example, if you're using something like, variational autoencoder model, you're basically able to interpolate the entire multimodal space, right? And so that can be a really powerful tool. We're working on, there's a student here kind of working on a project on that now. Where you obviously have to figure out how you want to navigate that space because you can't visualize everything, but all the parameters are accessible, right? And so it's just a question of how do you navigate that, and how do you kind of reduce it down in some ways. So yes, it adds an additional challenge, but that challenge also comes with some additional potential benefits. So
yeah, sure.
Yes, it's really important, I think. And if you're just interested in a black box prediction, well, you don't need that. But most of the time, we're interested in sort of what is the brain doing, or how what is this telling us about the disorder? And there you need to visualize.
Right. Thank you.
Thank you. I think I briefly spotted Dipanjan, who has been communicated with you.
Again,
he is actually in the eastern part of the country. We are in the western part of the country, and maybe his connectivity there, wherever he is not so good. Dipanjan, can you hear me? No, I don't think so. Maybe I'll ask him to ask you questions offline later because it doesn't look like we can reach him. Yeah. Any other question? Dr. Tuteja has joined us. Dr. Tuteja?
Yeah. Thank you, Professor Jain. Sorry I got late. But I understand the topic you see which Professor Wallen has addressed, is of immense importance. And the center which is heading on translation research, whatever work which we have been doing, and especially the biomarkers of the brain, which need to take further. I'm sure you see in times to come, you see IIT Jodhpur and your organization both can work together. It's a fantastic talk, and I congratulate you for the same.
Thank you. Thank you.
Yeah, thank you. We hope after all this COVID situation is over, we can invite you so that we can have a physical session rather than virtual session. Yeah, yeah. So the
Sounds
Panjan has
good. Sounds good.
disappeared. Yeah, I mean, it's a beautiful place to be, actually. It's a beautiful state. You love it here once you come. So, I would now request for the closing. Dr. Manish, would you like to do that?
Yeah, so this is a small token of appreciation from School of Aid, and we are going to send a physical copy of this, and soon this will reach you, and we
Wow.
hope you will like that. So yeah, and as Professor Jain said, we would be very happy to have you physically here and interact with you more on this topic. So thank you very much, and yeah, thanks again.
And maybe Dr. Toteza can tell a little bit more about what
Yeah,
it
sure.
is. Yeah.
Yeah. In fact, our foundation, Jodhpur City Knowledge and Innovation Foundation, which has been formed on the initiative of the Office of the Principal Scientific Advisor to the Government of India, has a number of verticals. We have the medical technology, we have the artificial intelligence, we have water and pollution. And then another vertical which we have been working is the craft. And this craft business is, huge business from India. We are exporting a lot, but especially in Rajasthan and further, Jodhpur, you see, it's a big business, and there are lakhs of people you are working from the unorganized sectors. They are so good, they are working from one generation to another generation and doing the things with their own hands, using the raw material available locally. So the one of the presentation, which one of this small token of the gift which we are presenting to our professor, who has kindly accepted it also, is a beautiful piece of the work by local artisans. So it is made of the camel bones. A lot of hard work has been put into it. It's a very artistical thing. And as Professor Jain said, that when Professor comes to India, he will see how these things are prepared with a lot of hard work. So
Great.
thank you, Professor, for accepting this small token of appreciation. And when you come here, I think you will see many more such items which are very useful and very artistically prepared. Thank you very much once again for your wonderful presentation.
Thank you so much. Is that a self-driving car?
We need to involve
That's very
Elon
nice.
Musk into this.
Yeah.
Thank you so much. Thank you so much for that. So yeah.
Okay. Thank you. Thanks, Vince.
It's a pleasure to join you all.
Yeah. Yeah,
Yeah.
thank you. Thanks for coming on. Yeah.
You're welcome. Okay. Take care.
Okay, we'll close it now. Okay, bye.
Thank you all.
Bye.
Thank you all.